Okay, so I think while people are still trickling in, we can go ahead and get started. It is my great pleasure today to have Yuhai Tu giving our physics colloquium today. I've known Yuhai for many years, but Yuhai has a long storied career uh, much before I came to the picture. So Yuhai got his PhD at UCSD in 1991, and he then spent 3 years at Caltech as a Division Prize Fellow uh, doing statistical physics research, and then he moved on to become a researcher at IBM Research in New York State, where he has been since. So it has been over 30 years, and over the years he has contributed to several different fields, some of which include statistical physics, biological physics, and so on. And recently, Yuha has also done a lot of work in machine learning and physics-inspired machine learning. So it's really a great pleasure to have here. Among his many honors, several fellowships, and so on, one of which that I want to mention is the Onsager Prize from the American Physical Society, which is the highest prize they have for statistical physics work, which is a great honor. So it's a real pleasure to have you here, Yuhai, and look forward to your talk.
Thank you so much. Let me. Great. That's the right move. Okay. So thank you, Navish, for that very generous introduction. So before I go on, let me acknowledge people. In particular, I think Howard Berg. Howard, I met Howard 23 years ago, 25 years ago, 2000, 2001, 24. And he's really the one who introduced me to the field of chemotaxis in particular, but in general, biophysics, right? The beauty of it and so on. And he also taught me something that's very valuable, that don't listen to anybody who tells you that certain problem is solved. It's never that way. So as a result, he's stick in the field since for his whole career in bacterial chemotaxis. Every so often, he discovered a wonderful technique to probe the system ever fine grain scale, and he discovered new physics and new biology, right? So that's the lesson that I really listen to that advice very. So since the time I met Howard, which is 2001, I have been working on bacterial chemotaxis, various part of it. So every time people tell me, "Are you still working on it?" I say, "Yes, absolutely. You betcha." And another nice thing about working with Howard is that I got the chance to meet wonderful postdocs, who later on become, PIs in their own institution, including people like, Navish, who we worked together with Howard, and we wrote probably the last paper with Howard when he was still alive. And by the way, also because of that, and Navish got to invite me to give a talk in this wonderful place. Anyway, enough introduction. All right, so the title of my talk sounds a little weird, right? Energy cost of biological function. People don't think about that, especially sort of a signaling or information processing in biological system. But as physicists who are interested in biology, certainly Howard was one of the pioneers, certainly not the first one to think about it from a physics perspective. This goes all the way back to people like Schrödinger. How many people has read this book or aware of this book? Okay, so it's pretty good. So I think a lot of the pioneers of molecular cellular biology, including Watson and Crick, actually know about this thing, this work, this paper, this little same book, and got inspired. So what Schrödinger was asking, what is life? What he really meant was the following question: how are living systems different from non-living or physical systems, which he's the one of the founding fathers of quantum mechanics. So he was very curious about how does living matter different from physical matter, right? Which he knows how to describe and using mathematical tools, Schrödinger equation, and so on. And in that book, he came to many conclusions, some of which turned out to be not so right. but nevertheless inspired people to work on the correct answer. But he made one statement which almost it cannot be wrong, right? It's kind of one of the statements that cannot be wrong. He made the statement that life feeds on negative entropy. By negative entropy, he really meant to say free energy. If you read his book, he actually write a little note that he used negative entropy because people understand entropy, but people don't understand the concept of free energy. So he used negative entropy as a way to say free energy. So he really meant this free energy. So life feeds on free energy. You can rephrase the Schrödinger's quote. In other words, life costs free energy. So that's a very broad statement, which cannot be wrong. Why? Because second law of thermodynamics say, well, if you have an isolated system, if you let it run long enough, entropy will take over, and you become disordered in the end, right? But life, you see order everywhere you see order. That's what life is. It's about order, not particular crystalline order, but order. You want your eyes to be there. You want your head to be above. So all the sort of from molecular to cellular to all kinds of different level, they are orders, right? So the central question really is, how do living systems process information? That I want to ask is that accurately with noisy component and stochastic reaction, to process information accurately leads to order, right? For example, in DNA replication, you have this base pairing, right? That's order, and that order is enforced by, of course, the base pairing. I will see that's the story is much more than that. And you have spatial ordering, right? You have the development in Drosophila, you get a spatial pattern. And you have temporal ordering. You have oscillation, you have circadian clock that oscillate in certain frequency. And you have high order, order, meaning like learning and information processing that happens in your brain. That all of this happens based on biological system that's quite noisy. The number of molecules involved in these biochemical systems are rather small, much smaller than Avogadro constant. So where does the order come about, despite all the stochasticity and noise and so on? So there are two questions that come to mind. One is, how does it do it, right? What is the molecular machinery and the mechanism and design principle that allow biomolecular system to achieve these functions that leads to biological order? Today, I'm going to bring a new perspective to this, is how much, right? So once you figure out a mechanism for this to happen, how much energy does this mechanism cost you, right? Or does it cost anything in terms of energetics? Sort of all in some broader sense, try to answer the question of the statement, try to embody the statement that made by Schrödinger, that life costs free energy. How does it do it? How much does it cost? Thinking about energy, you go open a textbook in Bruce Albert, a textbook about molecular biology of the cell. He actually tells you what energy is being used. Energy, of course, is harvested by biosynthesis, by photosynthesis, and it's stored in the currency as ATP. That's the source of energy for most biomolecular processes. So he said, well, okay, you have synthesis, you're building stuff, right? The building block is the material is there, but you want to put them together in a certain way, you need energy to put them together, okay? You have transport motility, you have all these molecular motors putting things around. Transport motility, of course, costs energy. That we understand, right? You have pumps and heat. Also, sort of all of these are related to mechanical motion or synthesis and so on. So, but what he didn't mention, which I think is the topic of this talk, is that what about information processing? Broadly defined as, how do you regulate things? How do you control things? How does memory be encoded and erased and rewrite? How does these processes, all about computing, that happen in your system, that actually, what is the sort of free energy cost for that, right? That leads to certain function that is not much to do with synthesis or transport, or all these known functional classes, right? For example, signal transduction, do you have to involve energy? Have you thought about, you have all these kinases which constantly turn ATP into ADP and the phosphate group that modify the the substrate so that you have different conformation, and constantly the dephosphorylation happen. So you have constant turnover of ATP using ATP hydrolysis. You have constant turnover of ATP. How do you count for that energy? What is that energy used for? Is it just if you create some other molecule, some signaling molecule that doesn't come with this energy of within ATP, would the system still work, right? So that's the kind of question we want to ask. So do you have example of that sort? So absolutely. So how many people have heard about John Hopfield? Let's just say John Hopfield. And because the Nobel Prize, right, last year. But okay, so if earlier 2024, somebody says, oh, you know, I bet John Hopfield is going to win Nobel Prize this year, and ask me that which work would he win Nobel Prize? Actually, I'm not so sure. As a biophysicist, I would say, well, maybe this work will actually be very important in biophysics. So anyway, so this is the work that I think could have won Nobel Prize. Of course, he won Nobel Prize for Hopfield model. That's a subject for a different talk. But here, I'm using this as an example of how sort of a system, non-equilibrium system, was able to do something that equilibrium system couldn't do by using energy in a clever way. Okay, so kinetic proofreading was motivated by the fact that if you look at base pairing, simple synthesis of, let's say, DNA replication by, you have a template, and you just want to build a complementary strand based on the template, right? So let's say you have one side that the correct base pairing is, the correct one is C, but the incorrect one will also form a stable bond, but that bond will have a higher energy. So so the equilibrium physics will tell you that, okay, given that differences in energy, you will have the error rate to be exponential e to the minus delta G being the free energy difference between the correct base pairing and incorrect base pairing, right? And that will lead you to a error rate of P. P is 10 to the minus 3, let's say, 10 to the minus 3 or 4, depending on the energy difference. That's far, far higher, much higher than what's observed. So that was the puzzle. So how do you create a system that replicates itself by making, much less mistake than an equilibrium model will tell you? So by and large, the mechanism, I should encourage you to actually read this paper published, 1974 in PNNS, where John Hopfield proposed a very generic model. Basically says, well, whatever the base pairing happened to be there, it comes and it forms intermediate state, right? C star C. And the intermediate state in the original model, in the equilibrium model, will just become a product with certain rate, right? And the incorrect one will also form the intermediate state and have the same production rate, but the binding and unbinding rate would be different for the correct and incorrect. And the ratio between the correct binding and incorrect binding, the ratio of that will give you e to the minus delta G. That's the error rate you're going to get the equilibrium model. So you say, well, okay, if you do that, you get e to the minus delta G. But what if you introduce another intermediate, say, CC, this is a triangle on top, that instead of going directly from there to product, you have another pathway to go back. To go back through CC, you go back to C plus C, which is unbind state, right? If you actually using energy to encourage that to happen, then you have a second chance, right? You don't immediately make product after you form the intermediate. You actually, so basically the picture that I have in mind, you could also do that, is that once you form the intermediate state, you test it. How do you test it? You test it by saying, okay, you do form a stable bound, but how stable you are. So the bound that formed by the incorrect base pairing is a little weaker. It's still stable, but a little weaker. So you just shake it. You just shake the bound with same amount of energy, let's say ATP. If ATP, the amount of energy injected to the system to shake it, is just above the binding energy of the incorrect base pairing, but still below the binding energy of the correct base pairing, then the incorrect base pairing will have a higher chance, almost certainly will fall off. but the correct one wouldn't. Well, it was much lower probability. In that way, you have a second chance, right? So by having a second chance, of course, just we do, if you have a second chance, the first one give you an error rate of p, small number 10 to the minus 3. Having a second chance will decrease the error rate by compound it, so it becomes p squared. So that's the basic idea behind kinetic proofreading, that you're going by introducing another kinetic pathway aided by ATP hydrolysis. So this, I didn't show it here. This process of going from C star C to CC, the red circle, is actually aided, is actually helped, driven by ATP hydrolysis. So that you actually, that's the shaking part, right? So as a result, so okay, so fast forward, this is the part of the work that not too many people know about, is that actually my colleague at Charlie Bennett, who's known for his quantum computing and quantum cryptography work, but early on he did work in biological system information processing, and he shows that yes, you can do that. I mean, Hopfield did know that, but Hopfield didn't compute the energy cost of this process. So Charlie actually did compute the energy cost of this kinetic. If you add this additional process in, it does improve the error. It decreases the error rate, but it also come with a cost. The cost comes with the two things. One is the energetic cost, and also you slow down. You slow down the whole synthesis process, right? So he actually come up with a dissipation error trade-off in kinetic proofreading in 1979. This is actually much less known paper. So what we have been doing since 2008 or so, late 2000s, are trying to sort of follow this kind of thinking, right? You first figure out how the system works, and then you figure out how much does it cost for it to work, and to relate the cost and performance, right? This function that you actually perform, how does it relate it to the actual cost? The cost performance trade-off is something that we have been working on for many different systems. So I mentioned the word non-equilibrium, right? So you say, what do you mean by being non-equilibrium, out of equilibrium? What do you mean by that? So of course, the best way to understand non-equilibrium is to understand what do we mean by equilibrium, right? So what is an equilibrium process? Well, this you don't have to go very far. I mean, you can go backwards even further. In 1931, Onsager, the Onsager Prize Onsager, he was very famous among physicists because he solved the 2D Ising model analytically. But he didn't win his Nobel Prize in physics. He won his Nobel Prize in chemistry, and for a work that's totally unrelated to the Ising model, which we did in 1940 during the wartime, right? But 1931, he writes this famous paper called Reciprocal Relation in Irreversible Processes, where he proposed, he actually showed if you're in an equilibrium system, time reversal symmetry is preserved, and you get this so-called Onsager reciprocal relation. That is, you have many input, x1, x2, x3, and you have the corresponding output, J1, J2, J3. And because things are coupled with each other, if they're coupled with each other through an equilibrium interaction, that means that if the L constant in that equation is basically the response function, right? Lij is that if I have an input of J, and if I measure the output at the x to I direction, and then you should get a response that proportional to Lij, right? So that's but what Onsager showed is that the matrix L is a symmetric matrix, okay? So Lij is equal to Lji. So why do I tell you all this? Because if you read that paper, I mean, I again encourage people to read this paper, very interesting paper, all the way down to chapter 3 of this paper. It actually made an analogy with chemical reaction. This is amazing. I mean, I don't know how many people know about this. So he actually made an analogy, say, well, okay, suppose I can make an analogy. Suppose you're looking at an analogy in chemical reaction in the sense that you have A, B, and C, 3 species, and between the different species, you have the transition rate. KAB corresponds to the transition from A to B, and KBA corresponds transition rate from B to A, okay, and so on and so forth. So if detail balance is satisfied, so detail balance is basically saying, well, okay, there shouldn't be any imbalance between the two microscopic state A and B. So the flux going one way should be going to the same as flux going to the other way if the system is equilibrium. You reach an equilibrium steady state. So that means that you have three relations, right? Because you have three links. So you have KBA times N correspond to the steady state probability of in state A, and so on. So you have three equations. So that's three equations for detail balance on all the links, okay? So, okay, what do I have to do with that? Then what do is you multiply all the three equations, right? So the multiply the three equations tells you that even if I don't know anything about N, I mean, N you need to solve. N depends on K, right? In the steady state. Even if you don't solve the whole system, if its detail balance is satisfied, then what I call the cycle rule has to be satisfied. So this is actually written in Onsager's 1931 paper, that in the equilibrium system, the product of all the rate going one way, let's say in this way, AC, CB, and BA, that's the counterclockwise, the product of all the rate going counterclockwise should be the same as the product of going clockwise, right? Once that is satisfied, your system is in equilibrium. It's an equilibrium, and the steady state you reach, the equilibrium steady state, which can be totally determined by the energy level of the three states, right? Okay, so that's the cycle rule, which we will use. Okay, now again, fast forward. This, I think, is somebody who's been my hero, one of my heroes in this business. I think he's really one of the, if not the one founding, but one of the founding fathers of non-equilibrium statistical physics or non-equilibrium thermodynamics. We talked about this with Rolf over there at the back, so he would agree. At least one people would agree. But what he said is that, look, this is fine, but in living system, you're often driven out of equilibrium. By which he meant that, suppose given the same system A, B, and C, right? And this original rate, K1, K2, K3, are modified because it's coupled. Some of the reaction are coupled to a energetically driven reaction. For example, in here, in the red line I show is a ATP hydrolysis. ATP is changed into ADP and inorganic phosphate. That's a low energy state, lower free energy state, and ATP is a high energy state. So by just sort of couple that with energy source, with an energy dissipated process, what you find is that, of course, then the cycle rule is broken. As a result, even if you reach the steady state, so this can still reach a steady state, meaning that the probability in A and B and C would no longer change in time. Even if you start with some initial condition, eventually the probability of in A, B, and C will be constant in time. But in this out of equilibrium non-equilibrium steady state, you have persistent current going around the cycle. Whereas in the equilibrium system with detail balance, the current is zero, right? So the persistent current J in the steady state, which can be written this way, is exactly, you can write it down, right? That's a very nice way to write it down, but I won't go into details. If you write it down, what you find is that in the denominator, those horrible 9 terms, they are positive definite, but in the numerator, it's exactly K1 times K2 times K3 minus K minus 1, K minus 2, K minus K3. So in equilibrium system, this numerator is 0. So therefore, there's no current, right? But in out of equilibrium system, in the steady state, J is not 0. And J has a directionality, right? So either going net counterclockwise and clockwise. So furthermore, what he showed is that because of this, you can also define a thermodynamic driving force, right? Delta mu, it can be computed, and it's KBT times the log of this ratio of the product for counterclockwise versus clockwise. And then, of course, if you have a current and the force, you can actually compute the dissipation rate, the rate of dissipation, how much energy is dissipated per unit time in the system. So you can just say delta W dot is a rate, is a power consumption. The power consumption of the system is J times delta mu. And you can easily prove this is a positive definite term, and it only becomes zero when the equilibrium cycle rule is satisfied. When delta J, delta mu, and J are zero simultaneously. That's the only way for it to be zero, and all the as long as you have a finite J, it's a positive definite. So of course, the system is out of equilibrium. Is actually you have to drive it with a dissipation that's provided by the external, in this case, ATP hydrolysis. All right. So in other words, continuous energy dissipation is needed to maintain a non-equilibrium steady state. So every time you see a persistent current, the system is out of equilibrium, driven out of equilibrium, you can actually compute the current, the persistent current, as well as the dissipation rate, right? So this is very important. So this sets the background for what I'm going to tell you next. So following this line of thought, and with all these techniques and ideas and concept that we get from the previous giants in the field, we have, since 2008, we have been doing a lot of work in different specific biological systems, try to connect the dissipation and with the function and the performance of the function. And in different biological systems, the performance is characterized by different things, as I will tell you. So I will tell you two examples, if time permits. One is about ultra-sensitive biological, biochemical switch. This is the paper published in 2008. This is where I entered the field, right? This is the very first paper I wrote about on the topic. And we have been working on this main theme for the last, 17 years or so. Yeah, so this is, okay, back to this case one. So this, don't mind Maxwell demon and all that stuff. So what I'm talking about here is the flagellar motor. So this is something that Namish and I worked together with Howard. So this is Howard's very word. So in 2003, he wrote this review article, which is really something started me in this direction of flagellar motor. He said it's really the bacterial flagellar motor is a nanotechnological marvel, right? This is really true because the size of the flagellar motor is the diameter is about 45 nanometer. And there are like 20 different parts, different proteins will come together, and the whole assembly process is just totally amazing, right? I won't get into detail. This is something very interesting. Another thing is that will be relevant to the talk of today is that the thing spins, right? Spins rather fast. It can be on the order of 100 hertz. The rotation speed is actually very fast. And on top of that, it actually can change rotational direction from counterclockwise to clockwise, and it can switch between these two. And the switching between counterclockwise and clockwise rotation is extremely important for the cell to look for better environment, to carry out not just swimming, but swimming for a purpose. And the purpose is to look for food. Okay, this is how it works, right? So basically, if the rotation is counterclockwise, it runs. It's swimming in the same direction. If the rotation is clockwise, it actually jiggles a little bit and change at the end of that jiggling, it actually orients in a different direction. So it changes direction and run again, right? So how would this allow you to search for food? Well, this, of course, have to couple with the signaling pathway, which again is a totally different topic I won't tell you about. But basically, what happened is that the signaling is controlling the duration of the runtime. So if my environment is pretty good or becoming better, then I will delay the switching of direction. So I will run a little longer. If at the end of my run, while I'm running, I'm sensing my environment becomes worse, then I will make my frequency of switching faster. So therefore, I will switch my direction, right? The switching itself is not directional, meaning that once if you decide to switch, you pick a random direction. And that's enough. By just controlling the runtime, to be correlated with your environment, with the gradient of the environment, is enough to allow the cell to actually go from a lower concentration of nutrient to a high concentration of nutrients. That's about the whole problem about chemotaxis is all that about. But today I'm going to tell you about the switching itself. So the switching is critical, right? The ability for allow it to switch, that is very important. So how do you characterize the switching? So again, so there's one sort of a very famous experiment done in 2000. So the switching, as I was showing before, is the switching frequency, which is called the bias, is determined by the intracellular signaling molecule KYP, right? So in this paper, they just changed the concentration intracellularly, changed it by using mutant that express this gene in different level. That's the x-axis. On the y-axis, what they show is the fraction of time you spend rotating clockwise, okay? So this is a response curve, basically input, and the output is the fraction of time, which we call the bias, right? The bias of clockwise bias is shown there. So what they find is that the response curve is very sharp, right? Has a Hill coefficient of 10, right? So a little bit of change of KYP will actually cause a huge change in the response in a very narrow region, of course. So this is actually a very well-known paper, and they coined the phrase ultra sensitivity because the sensitivity measured by the Hill coefficient is very large. So of course, the theorist says, oh, you know, based on the structure of what's switching, well, I can come up with a model to explain that, and this comes with a fancy name called conformational spread. So the ring that is the part of the rotor actually consists of a ring of Fly M, which actually combine with the signaling molecule, right? And this ring has 34 monomers form the ring. So if you assume there's some cooperativity between nearest neighbor of the Fly M, and you can actually come up with a model either based on Ising model that you have nearest neighbor coupling, or in the limit that you have very strong coupling, you actually come up with a simpler model called Monod, Wyman, and Shangzhu. Allostatic model can be used to explain here. You actually can show that the Hill coefficient indeed, H can be is proportional to n. N is number of subunit, which is in 34, times a 1 minus square root of c over 1 plus square root of c. And c is the ratio of the dissociation constant to the KYP for the counterclockwise versus the clockwise conformation. So the binding constant or the dissociation constant has to be different. C is defined over there. Oh, that's all fine. Okay, so that's not a problem anymore in somebody's mind. Problem solved. So don't listen to that. That's my. Then Cruzel did some other things. So I call it a spoiler because the spoiler to the sort of equilibrium picture of Ising model, MWC model based picture is being challenged by this experiment. On the surface, it may not be seen as a challenge, but it, you know, if you look deeper, as we did, it becomes a problem. It becomes a spoiler. Namely, what they did, Cruzel's lab, Philippe Cruzel's lab, what they did further is that instead of just looking at the bias, which is looking at the average value of the dwell time, right? They actually measure the whole distribution of the dwell time. And depending on, of course, the distribution will depend on the KYP level, which they did. So they're actually sweeping through the different KYP level, and they look at the distribution of this dwell time, and each curve corresponds to a distribution. Let's say the shaded one is actually clockwise, and the dark one is counterclockwise. Okay. So again, okay, they have find some distribution of the dwell time. So what's the big deal about that? So if you look carefully, you find, at least here, what you see is actually definitely for all this distribution, by and large, they have this peak, right, in the distribution. So that's also seems harmless. So okay, so what? You have a peak in the distribution. But that's a huge problem because then we say, okay, This is obviously measuring something about kinetics, about dynamics of the switching, instead of just the sort of the steady state switching, the response curve in the steady state. You're looking at the time, right? Time. So you have to look at the dynamic model instead of just a statistical physics model like Ising model or MWC model, which predict just the steady state behavior. You have to look at the dynamics. So okay, so we can come up with a dynamic model based on MWC model. For example, here, that the green dots are the counterclockwise, and the red dots are on the clockwise, right? So these are the two observable state. But you have an intrinsic state, which is how many QIP is bound, right? So then you have this extended square model that the ladder model, for example, you can call it, that on the horizontal lines are binding and unbinding, and the vertical line correspond to activation and deactivation, or the transition between counterclockwise and to clockwise or vice versa, right? So you can play this, you can put all the rates there. And if within the equilibrium model, right, then it has to satisfy the cycle rule. Remember that, right? So all the square, any square, the product of the rate going one way has to be the product of the rate going the other way. That's shown there. What I call thermodynamic relation, that's basically what cycle rule tells you. And that has to be satisfied for every one of these squares, okay? That still doesn't confine all the rates, right? You have, have n of these squares, you have n constraint, but you have at least 2n, you have 2n plus 2 rates, right? So it's totally, so you can play with other degree of freedom, still constraining by this. So we played with that, right? We actually did all kinds of combination of k's and omegas while satisfy this constraint. We never, ever get this to work. You can try yourself. I encourage you to do it. You can never get a peak in the distribution of the dual time. Never. Okay. Then that prompts us to think maybe this can never be done. But how we prove it? So okay. So this is, in my whole career, I'm a physicist. I don't prove theorem except this time. So I was able to prove that for all equilibrium system with detailed balance, the steady state dual time distribution can be written as the following form, right? It's the sum of exponential decay. But what I was able to show is that A, the coefficient A, is positive definite if detailed balance is satisfied, right? So which means that if the detailed balance satisfied, if in equilibrium system, this function should be monotonically decreasing, like a Poisson distribution, right? And so on. And also, furthermore, it has to be convex, but that's a higher order moment, which is harder to show. So therefore, this is what the part of the paper is about, is that, okay, from the distribution, if the measurement has no problem, then I know the underlying mechanism cannot be an equilibrium model, including Ising model or MWC Eller-Stirk model. It's not correct. At least you have to modify it in a certain way. So that's basically what we're saying. So now we say, okay, can we modify the model in some mild way, in some simplest way possible that breaks detailed balance, but result to this peak, right? Can we see this peak? So this is a necessary condition, but not certainly. Basically, we're saying we ruled out equilibrium model, but we don't know the non-equilibrium model will actually generate this peak, right? So the question is that, can you construct a non-equilibrium model that will actually generate this peak, right? So this is the equilibrium model, but we can modify, again, like I said, what you do in non-equilibrium model is that you base on the equilibrium model, all the rates, then you actually selectively modify some of these rates by a factor, I call it gamma, right? As long as gamma is not the same on the same link going forward and backward, you break detailed balance. So that's what we did based on that. But we have other ideas of where do we put it in order to generate this function. Okay, so it turns out, so I don't know what drive the enhancement of these rates. So I call it Maxwell's demon just because it's a mysterious mechanism that actually do the job, but it also causes energy. Okay, so I can create that. And what I find is that if you choose your rate enhancement on the one side, lower QIP level, that going up, that you enhance the rate from going from clockwise to counterclockwise when QIP is, the bound number is less than some number, and you enhance the downward, right, the transition rate on the other side, then you separate the gamma, right? Then you are driving in a non-equilibrium fashion. And in that way, can I get a peak? So you generate a flux that's a signature of being out of equilibrium. And sure enough, you can, depending on the driving. So when gamma equals 1, that's the equilibrium model. You get a monotonically decreasing function. As you increase the driving, you increase gamma, right? It first form a little bump, and then the value at tau equals 0 get drive down, and eventually when the driving is very, very large, then you actually form this peak, single peak, right? So you can. That's just sort of a existent proof that indeed, by putting energy to drive certain rate going, increase certain rate, you actually can, right, have these statistics of dual time. So, but non-equilibrium steady state costs energy, but how much energy is dissipated? Then here comes Terry Hill, right? So we have figured out a mechanism to generate this, this peak, and we have this rate on all these transition. You know, Terry Hill tells us exactly how to compute this dissipation rate, and we did that. Basically, remember the J times delta mu, right? You just compute for every cycle, and you sum them up. That's how you calculate the dissipation. So on the horizontal, so here's what we did in the non-equilibrium. So remember, we started the thing by, I started the thing by showing you this high sensitivity, right? High response sensitivity, which is another observation that we want to match. So the question for us is that, okay, now the equilibrium mechanism, IZ model, MWC model, has a nice feature that it will have this high Hill coefficient. But it depends on the binding constant to be different, right? So if the binding constant is the same, then you don't get any response at all, right? So based on that no response equilibrium model, if now I inject energy in to push certain rate up on different side, can I generate a high response Hill coefficient? And the answer is yes, you can. So gamma, again, remember, is the measure of non-equilibriumness, log of gamma. So gamma equals 1, it means the equilibrium model. And in this case, we look at C equals 1, right? There's no binding difference between the CW and counterclockwise and clockwise. And as you see, as the driving goes up, as gamma goes up, the response curves start to generate a higher and higher response. And you can calculate based on Terry Hill's formalism the actual dissipation per switch, right? So the Terry Hill the formalism tells you the dissipation rate. If you multiply by the time, the average time of a switching, that gives you a delta W, which is per switch, how much energy does it cost you? And as you see, as you pump in more and more energy in the right place, you see the Hill coefficient start to go up, right? And it saturates. And the saturation point is determined by where do you put the flux, right? So if you the increase and the decrease and the increase on the both sides, the separation is delta n. If you separate them further and further apart, you actually can reach the saturation, higher and higher saturation value, okay? All the way towards the actual maximum. Okay, so that's story number 1. And of course, we have a mechanism, but the mechanism involves some increase of rate on one side, increase of the rate opposite on the other side. So where does it come from? Where does the molecular mechanism of the enhancement of these rates? So of course, the first thing people are skeptical is, experimental error, blah, blah, blah. So a paper by another former lab member from Howard's lab, Junhua Yuan, who has his own lab now back in China, actually roughly 10 years ago, actually have shown that indeed this actually peak happens, but it depends on the driving. So this is also another very clear indication that this is a dissipative property, because when you change the PMF, this peak might go away. Okay, so that's what's being shown here. So really the hunt is on. The demon is, so we have some hints of what the demon is. So we want to know what the demon is. What constitute this mysterious molecule or complex that actually can observe things and use energy to do things according to the observation. And so we know the driving force is probably PMF, and it's using mechanical force to control switching between counterclockwise and clockwise. And the hunt is on. This slide was written about a year ago. So right now we have a very good candidate of what are these demons. I think the demons are the stator, so which Navish and I worked on with Howard. And the stator have other wonderful properties, as well as I think also serve the purpose of switching, helping the switching, to enhance the switching cooperativity. Okay, so that's story number one. I don't know how much time do I have. Okay, so this is probably good enough to show another example, interesting example, just storytelling, right? So another system that we worked a lot on is circadian clock of the Chi. So Chi system is the cyanobacteria has this very nice property that it has a clock, has a 24-hour clock. And this clock also has the nice property that it consists of all other clocks, circadian clock, all involve gene expression. Right? This clock is a pure protein clock. So Kondo in 2005 was able to reconstitute the clock in a test tube by putting 3 purified proteins, Chi-A, B, and C, together in a test tube, and this is what you see in vitro, the oscillation of 24 hours, okay? But of course, you have to add ATP. You cannot, the clock wouldn't work without ATP. And the detailed mechanism, I will probably not show you too much. Basically, the clock works just like our clock. We have the clock, we have ticks, right? We have 12 ticks, right? Larger ones. The clock, the Chi-ABC clock also have 12 ticks, but ticks, each tick is a phosphorylation. So there are 6 monomers. This Chi-C forms a hexamer. In the C2 domain, the C1 and C2 domain, in the C2 domain of the monomer, each monomer have 2 phosphorylation sites, right? The S and the S431 and the T432. 2 tick, right? And you have 6 of them. You have 12 ticks. Basically, during the day, right, the phosphorylation go from 0 to 12. And during night, dephosphorylation happen. It goes from 12 back to 0. That's the day and night cycle. So basically, it's like this, right? So 12 a.m. the nothing, no phosphorylation. 6 a.m. you have 6, and then 12 p.m. noon you have, and then the cycle goes on. Okay. So we have come up with model to explain the cycle, but that model, you can see, well, okay, so we worry, so there's a mechanism to do that, but there's also a energetics to go along with it. So you can just count number of molecules, right? So this thing, of course, depends on phosphorylation. So during the day, each side gets phosphorylated. During night, it gets dephosphorylated. Of course, you cost you 1 ATP, right? Since you have every monomer have 2 phosphorylation sites, so every 24 hours, each monomer will cost you 2 ATP, right? That's how to drive the phosphorylation dephosphorylation cycle. Now, that's fine, that's good. And we have a model that based on that seems to be the right number in terms of energy cost, but they did the experiment. They actually did the experiment by counting how many ATP molecule was actually used during 24 hours for per monomer, and they find 16. So where did the other 14 go? What is it used for? There are many reasons for it to be, there are other sides, because the C2 domain is the phosphorylation dephosphorylation. There's a C1 domain, which is actually ATPase. Its function is totally unknown. So it probably goes through there, right? But what's the function? Does it have any function or just wasting any energy? So then we start to worry about synchronization. So if you think about it, it's actually a pretty serious problem, right? if you think about per hexamer, and going from one tick to another tick is phosphorylation, and this is a single molecule. There's a huge amount of noise, right? So you're looking at on average, the period maybe is 24 hours, but there's a huge fluctuation. Not only in terms of fluctuation, but in terms of the phase. If there are two molecular clocks are independent of each other, they will go into, on average, have the same period, maybe on average, but their phase can be totally out of sync. If you have thousands and tens of thousands of these hexamer not talking to each other at all, and they all oscillate very stochastically with the same period, once you average over all of them, which is what you measure, you get nothing, right? So you absolutely need some mechanism of synchronization between these molecular clocks for it to actually work, for you actually to explain the macroscopic behavior which you measure, right? So where does that come in? Okay, so now here's another model. Say, okay, they do communicate with each other. So what it's showing here is the torus. It's the in the phase. So the two direction, the torus, corresponds to the phase of 2 clock, right? 2 pi. So on the right-hand side, you have 4 dots, right? If it moves in one direction horizontally, that means the progression of 1 clock, processing of 1 clock. In the vertical axis is the precession of the other clock, right? So the red line corresponds to they talk to each other, right? So they actually exchange some phase information. So this is going to the realm of Kuramoto model. So basically 2 o'clock, if their phase is not the same, you have some kind of interaction to allow them to synchronize, to actually take the average. So the more advanced one will actually slow down a little bit, and the less advanced one will accelerate a little bit by this phase exchange interaction. So this is all theory. Okay, at this point, we just want to see whether this works. So this is a toy model, and we put it in, and this toy model depends on the exchange interaction, which is a chemical reaction. We model as a chemical reaction with a 2 arrow, right? So you have forward and backward rate, and the forward rate and backward rate satisfy local detail balance, meaning that the ratio is just the e to the minus delta g between the 2 states. That's delta E ij, right? And so we have a capital omega, which is the frequency of this exchange interaction, and delta E gives you the strength of that interaction. So this exchange reaction is characterized theoretically by these two. One is how frequently it happens. Another one is how strong, when it happens, how strong it is. So there are 2 parameters which you can use. But one thing I want to show your attention is that if you go around the big circle, meaning that one molecular clock go 24 hours, right? You got the cycle rule broken. That's the cost of progression of 1 clock. If you go the other direction, right, you get another big detail balance. You have another cost of individual clock going one way or the other, right? But by the introduction of the exchange interaction, the red line, you also have the local circle, right, cycle, because the existence of the red line. See that gamma L, right? Because of introduce that, so that also causes you energy, right? So that's sort of the pretext for actually what I'm going to tell you next is that, okay, by introducing this mechanism, first of all, it works, right? So this is a phase diagram. So remember I said this exchange interaction is characterized by how often it happens and how strong it happens, right? So if you have it more frequently, have it more strongly, you drive into the synchronized phase. If you don't drive it at all, it goes to the asynchronized phase, right? Which you don't see any oscillation, collective oscillation at all. So you can do that. But on top of that, we can also compute the dissipation, the cost of having the omega and having the E0. So in the E0 and omega plane, so like this, right? So this is, so you can actually compute the dissipation. And the dissipation consists of 2 parts, as I told you, that for each individual clock being able to run around, that's the 2 ATP they cost, right? But there's an additional cost because the local cycle, the synchronization cycle. That's the cost in the red part. And that's the cost that was shown here, the coupling energy cost down here. It's called W exchange. And what you find is the following, that yes, so on the y-axis is the order parameter here of synchronization. So if you're out of phase, no synchronization, this order parameter is 0. And you see the phase transition from asynchronization to synchronization happens at a finite value of energy cost. So it costs you to synchronize, right? This is on top of driving each individual clock. We believe this is the reason it costs more than the 2 ATP. So this is some theoretical result. But I think also another thing that you can look at it is that another question you can ask is that, okay, you remember I showed you this in the omega E0 space, the two characterization of the exchange interaction, the frequency and the strength, right? And the cost depends on these two parameters. You can ask the question, if I want to create a certain synchronization, what's the kind of combination of omega and E0 that costs the same amount of energy but doing better in terms of synchronization? So you have a design problem. So it turns out a dotted line corresponds to the best design. So you achieve the same level of synchronization with the minimum cost. Right? That's the dotted line. That's the sort of the perfect optimum design line. All right, is it really happening in real biological system? The answer is yes. An independent experiment in 2006, so this is actually before they realized this discrepancy between the ATP cost and actually the cycle itself. They did another experiment. They have 2 batches of the hexamer, right? One batch they actually attach to a linker, which can be used to pull down. So this is this batch, right? The left side. And then at time t equals 0, they mix them. They mix the 2 batch, right? If they don't have any exchange between the 2 populations in terms of monomer shoveling, then when you pull down, you only get half of the population. Right? But when they do the experiment, depending on where you do the experiment, actually you pull down 100%. You basically, there's some monomer shuffling. So the hexamer doesn't stay still. The energy is being used to actually disassemble the hexamer and reassemble it again. Disassemble, reassemble it again. And the frequency of that happen is our big omega. The strength, the E0 is something we don't know, molecular mechanism. So based on that, we actually have built a model which not only introduced the progression of individual clock go around the phosphorylation and dephosphorylation cycle, but we introduced the monomer shuffling part, that 2 clock with this different phosphorylation level can mix and becomes 2 individual clock which have different phosphorylation level. And by introducing that alone, you see that when the shoveling rate R increases to a level of 1 per hour, you actually start to have synchronization, right? And this is on the right-hand side, what you plotted here is the cost. So the bottom line is the 2 ATP, the cost for each individual clock to run, right? And it rises up as R increases. And the cost is because you actually constantly have to disassemble and constantly have to reassemble. And that's a costly process, but it's a worthwhile cost because it allows you to synchronize, allows you to communicate. Okay, last slide. So I think what we have been doing for the last 17 years is really trying to answer Schrödinger's question in some sense, in broadest way. Try to establish a non-equilibrium physics for living system. Out of equilibrium is something people know about, and Terry Hill and all knows about it. But biology is not just about that. That's only a necessary condition, that to realize that it does cost energy. So as Schrödinger realized that it has to cost energy. We agree with that. But energy is not sufficient, right? So we show that energy is necessary but not sufficient. So you need the clever way to design the way to inject, not design. There must be clever way by evolution, probably, surely, that actually the certain things are evolved so that this energy is not wasted. It's being used to enhance your function in the cases that we discussed. But in some other cases, it actually, what I show here is adaptation. Adaptation, as we're telling people during lunch, could never happen in an equilibrium system. If I prove any theorem, that's the second theorem I will prove. I think we sort of proved that, but anyway, so let me end there. I think living system as an information processing engine and machine is a very interesting thing. And in the spirit of Carnot, when he looked at the heat engine and look at the efficiency and the cost and performance, the work done, what we do is to try to understand this information living machine in living system. What is the efficiency in some sense? The efficiency is no longer about the actual mechanical work. It's about how good you do signal processing, how accurate you are, how fast you are, and how much synchronization you create. Okay, with that, I thank you for your attention.
Maybe one or two quick questions if anyone has them. So I have a question regarding the hexamer example.
Mm-hmm.
So you're saying there's the two hexamer plates chain.
Right.
So does it always happen parallel, or there's always like?
No, we actually do. I mean, now my collaborators are doing experiment to exactly understand the process. It definitely that's not pairwise. It's not like monomer shuffling. Sort of a cartoon picture is that they come together and they change polymer. I think the more likely picture is that they disassemble in a very arbitrary way. They could be former, partially formed, and two dangling around. And so that has a lot to do with the ATPase activity, because the C1 domain, the ATP activity happens between two adjacent monomer. If that goes through ATP hydrolysis, that link becomes loose. So if you have one loose, it wouldn't fall apart. But if you have two, then it falls apart. Before it rejoins, it falls apart. So so we have now a much more mechanistic way of understanding how the shuffling happens. So it's definitely not pairwise. The way we model it, yeah, that's pairwise, but that's not the true mechanism. I'm just showing what we're trying to show there is that as long as you have some exchange, even a simple pairwise exchange, you will actually create the synchronization through that exchange. But that process costs energy. So that's the main point. But detailed mechanistic molecular mechanism is surely not, and we know for sure now, it's not what I just showed you, that only two collide and exchange and goes away. Right. Right.
It looks like three of them are bundled, two are unbundled.
Yeah.
So the question is really two states, because the whole argument of detail balance versus not detail balance was, if you have exponential weighting in each state, you can't tell whether you're detail balance violating or not. But there could be a whole series of latent underlying states.
Yeah. Yeah, so that's what basically we're trying to do here. But remind you, one thing is that most of the experiment I showed you is for one flagellar motor. That's nothing to do with interaction between multiple flagellar motor and most of the experiment were done by shearing the flagellum and attach a bead to it. Yeah, so that's a good question. So we did thought about that, but as long I tell you this, you can have hidden state as we're showing you, as long as it's equilibrium, the hidden state transition between hidden states satisfy detail balance. You will never get this duration time for the two observable to have the distribution. Yeah. So it's about the hidden state and the fact that the hidden state allows you to introduce this daemon. Daemon will act on this hidden state. So what we find is now is that we actually have a way of, understand how this happened molecularly.